All right, thank you everybody for joining us today. This is the Science and Innovation Center webinar, part of the webinar series that was originally planned for ATS in May and is now being done virtually. Today, sorry about that. So today's webinar is part of the Science and Innovation Center virtual ATS series. My name is Carrie Piston. I'm the moderator. We have three speakers today who I'm excited to introduce. Stephanie Christenson from UCSF, Craig Hersh from Brigham and Women's Hospital, and Pratik Sinha from Washington University. A few other reminders. We have another seminar coming up in 2 weeks on executive coaching, also from the Science and Innovation Center. And then this is part of ATS Virtual, and you can still register. All of the content is available through the November 10th. And then all of the SIC webinar videos are archived at this link, and this one will be too when it's completed. And we're also looking for feedback, so please take a moment to give that at the end. So briefly, just an introduction to today's webinar and the topic, which again is talking about getting to endotypes in the study of complex diseases. Complex diseases are multifactorial, generally thought to have genetic, environmental, and lifestyle risk factors, and they're often heterogeneous. There are multiple phenotypes, which are observable traits or characteristics, and these can also change over time. And there are also thought to be multiple endotypes, which are the biological mechanisms responsible for the disease pathology. And these endotypes don't necessarily line up well with the phenotypes, which can make understanding disease etiology and mechanisms quite difficult in the complex diseases. So the goals in general are to identify endotypes, goals of, I'd say, some research or this topic in general, to identify endotypes, and then number two, to then study these subgroups of the complex diseases in order to improve our ability to identify therapies. So our speakers today have done really exciting work in this field. So first, I'm going to introduce Stephanie Christenson from the University of California, San Francisco, who's going to talk about genomic profiling of airway samples in asthma and COPD.
All right, so let me share my screen. All right, so thanks for that introduction, Carrie. Yes, so I'm going to talk about genomic profiling of airway samples. And so my, let's see here. Here are my disclosures, but nothing really pertains to, none of my consulting work really pertains to my research. So what I do in my research is I really study molecular phenotypes, or I'm going to use that word kind of interchangeably with endotypes here, although they're not quite the same thing. But I study molecular phenotypes using RNA sequencing or microarray transcriptomic methods, mostly of airway samples, so meaning airway epithelial cells from brushings, or sputum samples, tracheal aspirates. And I'm going to talk about this mostly in the context of airway diseases, so asthma and COPD, but I also work in other diseases like ARDS and pneumonias. I work with Pratik's group some in their lung injury work. So a lot of what I'm going to talk about today can really be used for other diseases as well. And so I'm going to really kind of focus more on methods. So I think Carrie gave a great introduction to endotyping or molecular phenotyping. And so I'm going to talk a little bit about how we've built immune response signatures to study endotypes or molecular phenotypes. I'm going to talk a little bit about how cellular deconvolution can kind of aid in understanding or improving the depth of biological understanding of some of these phenotypes. And then a little bit on metatranscriptomics, or understanding how bacteria, viruses, other microbes interact with host gene expression using RNA sequencing techniques. And so these are some of the folks that have really contributed greatly to this work, along with others who I'll get to at the end. All right. So I'm just going to kind of breeze through this because I think we all understand that many of our lung diseases are really complex and heterogeneous. And we see that clinically. COPD is a great example of that, and that not everybody has emphysema or lung tissue destruction. Not everybody has chronic bronchitis or chronic cough. And some people have some mix of these, and we don't have great therapies, meaning we don't have great therapies that target everybody. So we study this really in the context of these molecular phenotypes. So, and I don't have this is not an exhaustive list of molecular phenotypes, and meaning potential groups that have a particular type of biology underlying their disease. But the reason I bring up the ones that are listed here are that we have therapies that target these things. So particularly when we think of type 2 inflammation in asthma, we certainly have biologics targeting that as well as inhaled steroids. But these other ones as well, you know, a little less known in airway disease, but there are other places where we treat some of these issues. Okay. So we're first just going to talk about how I identify molecular phenotypes using the transcriptional signatures of these adaptive immune responses. And what we've really focused on in asthma and COPD are these kind of polarized adaptive immune responses that are really kind of the big ones. So type 2 inflammation, so if you think of eosinophilic, the classic allergic response, type 17 or type 3 inflammation, which I usually think of as antibacterial or targeting a bacterial response, usually driven by IL-17 and associated with neutrophils. And then the type 1 response, which would be kind of an interferon-driven response. So usually thought of as more of an antiviral response. And so we've looked at all three of these and have generated signatures for all three of these. And I'm just going to show you a little bit about how we've done that. All right, so first of all, taking a step back, a lot of this approach has come from my mentor, Prescott Woodruff at UCSF, along with his mentor, John Fahey, many years ago, along with the group at Genentech, where basically they took, they did research bronchoscopy, where they went down into the airways of steroid-naive asthmatics and healthy controls, and they grabbed airway brushing. So just grabbed a brush, and then they took that brush and ran it on microarrays. This is before RNA sequencing. And what they found was that when they compared asthma versus health, that most of the really highly upregulated genes were really upregulated in a subgroup. And so this kind of subgroup that you see here in red, which are all asthmatics, but then you also see low expression, that's healthy controls, all the healthy controls belong there, but so do a lot of the asthmatics. So this was kind of one of the first molecular phenotyping studies using gene expression. And what they found was that this group of asthmatics was the Th2 high subgroup, meaning this was the group that had more IL-13, IL-5 inflammation and responded better to steroids. Now, the issue with this approach is, you know, this is great. If you can find that you have a molecular phenotype just based on healthy versus control, and then you do a little clustering and you get the phenotype, that's wonderful. But that does not work all the time. And in fact, doing the same type of approach in COPD doesn't work. So in COPD, we're very interested in type 2 inflammation or eosinophilic inflammation because we're really interested in the asthma COPD overlap population, meaning the group that kind of behaves more like asthmatics, but they're still COPD, because we think they could potentially be treated a little differently. So to look at that group in COPD, I can't just use those three genes these three genes here, periostin, CLICA1, and serpin-B2, because they're very smoking responsive, and really, we don't want to just measure smoking. So we ended up taking the 100 genes that are most upregulated in this Th2 high subgroup. So this kind of signature of 100 genes from the Th2 high, and we use that to train the COPD subjects. So we had kind of the same set, or a similar set of data set where we had bronchial epithelial brushings in COPD and healthy control, as we did in asthma, where we had asthma versus healthy control. And we used the 100 genes and we put it into this PCA projection algorithm, which I'm not going to get too much into the details here, but you can look at our Blue Journal paper on it, where basically I used that asthma data to train the COPD data, and then we could take that and we could use a signature that I created from those 100 genes matched to the asthma data, and I could create this metric, this type 2 score. So telling me if you are potentially type 2 high or type 2 low, and that kind of on a gradient. And what we found was that this signature score really did seem to measure an asthma COPD overlap group well. So it was associated with increased eosinophils, increased bronchodilator reversibility, and improvement in hyperinflation, meaning how much air trapping you had in response to steroids, meaning that if you got steroids, that you maybe had a little less inflammation in your small airways, and that improved your ability to take a breath out. So it really kind of looked like a asthma COPD overlap group. So using this signature, now we can identify using these airway samples, who is type 2 high and who is type 2 low. Okay, so I'm going to just go on, and again, I'm not going to get too far into the biology or clinical implications, but kind of trying to understand some of the other methods used. So now the next one we did was looking at IL-17 inflammation. And IL-17 in COPD we think of as very interesting. So IL-17 is more of an antibacterial response or a neutrophilic response. And we think of COPD as, classically it's thought of as sort of a neutrophilic disease, or at least that there's a subgroup that potentially has high neutrophils. So we were very interested in understanding who might be IL-17 high. And to actually identify that subgroup, now I didn't have asthmatics to go after and try to understand who is asthmatic or not, or who is IL-17 or high or not. So instead we used experimental data. So basically what we did was we had our colleagues here at UCSF, Luke Bonser, do air-liquid interface experiments for us of human bronchial epithelial cells where they stimulated with IL-17, and then they did isolation of those cells and ran them on sequencing. And then we could identify the cells or the genes that were most upregulated in association with IL-17. Okay, and so with that, then I can take those genes. So we would take like the top 100, top 200, depending on what you're looking at. Here we took the top 100. And I basically took that list of genes and I took it into another data set, a COPD data set. So actually this is one of the same ones that I used to generate the type 2 signature, where we have human bronchial epithelial cells that we got from research bronchoscopy. And I was trying to identify in these samples which genes really clustered together, meaning that it looked like they were all in the same pathway and kind of all moved together, so tight clustering. And so basically using that approach, we identified this 11 gene IL-17 signature. And so now I was, as opposed to the asthma where I'm like, well, I think, we know that these are asthma genes. Here, it's still a little unclear, are these IL-17 genes? And so we can actually look to public data to help us understand that. So we did that. So actually IL-17 is a target of therapeutics for other diseases. So it's a target for psoriasis-based disease, so basic psoriasis and psoriatic arthritis. And there's actually data sets already available where there's been IL-17 blockade. So we could look at that where we looked at skin lesions where the patients had psoriasis and then got IL-17 blockade, and we could see what our signature did in response to that. So here is one where they got ixekizumab, and if they got placebo, nothing really happened to the IL-17 signature, but if they got ixekizumab, it went down over time. And then this is just another one where patients got brodalumab, which is another anti-IL-17 medication. And what this shows is that if you don't have a lesion, you have a low, the signature is low. If you're IL-17, or if in the psoriasis lesion, the IL-17 signature is high. And then at weeks 1, 2, and 6, we actually see somewhat of a response to the signature in the setting of brodalumab. So at week 1, and that's really with these 300 and 500 milligram therapies, which is actually similar to clinical response. So If you got placebo, you didn't have improvement in your psoriasis lesion. And if you had the low dose of brodalumab, you didn't have improvement in your psoriasis lesion. But if you got the higher doses, you did. So we see the IL-17 signature going down over time when you get brodalumab. So it really suggests that our signature is actually measuring an IL-17 response. And then when we take it back into other data sets, we can see, okay, what is this associated with? So now I can measure the IL-17 response, and we see that it's associated with neutrophils and macrophages. And it's actually also associated with a decreased response to inhaled corticosteroids. So kind of like the anti-asthma COPD overlap response, meaning that if you get an inhaled corticosteroid, you're unlikely to have a response in your FEV1 if you are IL-17 high. Okay, let's see, how am I doing on time here? Okay, so a couple other things that we'll just kind of touch on briefly, and that's one of those is cellular deconvolution. And this method, basically, we will let single cell data help inform our understanding of what's going on in the bulk sequencing data. So meaning that we take signatures from single cell sequencing data. So in this case, this is actually an example from severe asthma, where we had endobronchial biopsies taken from the airway, and single cell sequencing was done. And we identified the cell-specific transcriptome. So we could identify goblet cell signatures, dendritic cell signatures, macrophage signatures. So what is the specific response of, or the specific genes that are associated with the specific cells? And then we can take that data and take it back to the bulk sequencing data. So by using these kind of cell-specific gene expression signatures, we can understand what is happening in our sequencing data. And then I can actually relate that to my inflammatory signatures. So what you see here is a dot plot, and if you can see that the type 2 signature, which I have highlighted there in the rows, is really associated with having an increase in goblet cells and mast cells, as well as ER stress. Whereas IL-17 and interferon signatures are really more associated with inflammatory dendritic cells and macrophages and neutrophils. So kind of what we would expect to see in associations here, but it's kind of interesting that we add this extra depth of what are the cell types that seem to be upregulated in association with us. All right, and then actually I'm just going to skip through this really quick just so that I don't take too long. The next thing that we did, and this I'm just going to really briefly touch on, is that we actually use metatranscriptomics. So actually understanding not just what's present from the host in this bulk sequencing data, but also what is potentially coming from viruses or bacteria or fungus in these same samples. And we can use that to understand a little bit more about the molecular phenotype. So in this case, we were studying exacerbations of COPD, where we know that all exacerbations are not created equal. Some are due to viruses, some are due to eosinophils or type 2 inflammation, some are due to bacteria, and some are due to actually other things like PEs or heart failure exacerbations that are kind of masquerading as exacerbations. But we have a hard time treating exacerbations, and we tend to throw the kitchen sink at them, where everybody gets steroids and antibiotics, and that doesn't always seem to make a lot of sense in the setting of the different types of exacerbations. So we can actually take metagenomic sequencing, where basically we take all these samples and we get rid of all the host data, meaning we really try to get rid of anything coming from the human. And then we will align to viruses and bacteria and fungus. And so we actually have been doing this for quite some time with folks at the Chan Zuckerberg Biohub, part of UCSF. And they actually now have an open resources called IDSeq that people can use. It's a free tool that's cloud-based that people can use if they are interested. And I'm not going to go through this because it'll take too long, but there's actually up on bioRxiv at the IDC paper. You can go through how they do the alignments. But using this, we are able to look at exacerbations. And so these are actually about around 43 exacerbation samples, and where we had measured interferon response, type 2 response, bacterial response in sputum samples. And then we could also identify whether they had virus, bacteria, or none of those things. We actually had eosinophils, so we can measure eosinophils as well. And so what we see here is that the viruses, which are in green on that color bar at top, are associated with a high interferon response, and showing that as a box plot. You see that that is true, that they're associated with this type 1 score. That looking at eosinophils, which are in pink at the top, are really associated with a type 2 score. And again, showing that in this plot here. And that bacterial exacerbations are really associated with a type 17 score. And again, showing that in the box platform. So basically that we can use the meta-transcriptome to look at host microbe interactions. And we've done this also in ARDS and in pneumonias. And so there's this technique can be used to really study host microbe interactions kind of throughout. All right. So the conclusion's really just that we've used several different tools to help us really understand molecular phenotypes. So inflammatory signatures, publicly available data that can assist us in validation of our findings, like these anti-IL-17 therapeutics. We use cellular deconvolution to really understand a little bit more about the molecular phenotypes and what kind of cells might be present. And then using metatranscriptomics and host microbe interactions to really understand really what else is in the microenvironment and how that might be influencing what the host is seeing. And there's a lot of people who have done this work, including folks from UCSF, folks from University of Groningen, the SARP investigators, the Spiromics investigators. We use a lot of multi-center data. And this is just our group at UCSF, at the Airway Clinical Research Center. And we've got a lot of interesting work going on there in case anybody is interested in training with us. All right, that's it.
Thanks so much, Stephanie. You guys, everybody can send questions through the chat. So if there's anything that comes through, but I was going to ask Stephanie, how are you using all that? Like, what are the next steps? Are you doing any or thinking about planning studies related to therapies? You mentioned the inhaled corticosteroid resistance in the high IL-17, or those were studies in psoriasis, right? Not in asthma? Those were studies.
Psoriasis. I will say that there are multiple companies that are very interested in anti-IL-17 therapies. You know, I'm not a trialist. You know, I'm more interested in the kind of genomic side, but we do help companies try to design their studies in ways that are thinking about these phenotypes, so that we might be able to find groups that have better targeted therapies. So, some of our work is really kind of focused on that, like understanding who's going to respond to therapies. Some is also looking at, can we identify these subgroups using less invasive techniques like blood or nasal swab, now that everybody and their brother is getting a nasal swab for COVID-19. There's other ways that we can potentially identify these things, because not everybody, I wouldn't want everybody to get a bronchoscopy to be doing molecular phenotyping. So that's some of our work going forward.
Great, thank you. So, trying to successfully share my slides. So this is just a slide where I had all the speakers lined up. So thank you so much, Stephanie. That was a really great first talk. And now I'm going to introduce Craig Hersh. I'm lucky to have as a colleague, who's going to talk about clinical and genomic subtyping in COPD and asthma and COPD overlap.
All right, thanks. Let me share my screen. Okay, can you see that? Okay.
Yes.
Okay, good. Thank you for having me, and thanks everybody for listening to this webinar. Let's So let's do this. So these are my disclosures. I think just to note that some of the IgE levels, which I'll talk about later, were funded by Novartis. So I just wanted to get into definitions a little bit. I think Stephanie gave a great talk, and she said she used the term phenotype loosely. So I'm going to use what my definitions are just for the rest of this talk. So in the classic genetics paradigm, phenotype is any observable characteristic. So in lung disease, we can think about things like FEV1, IgE level, a diagnosis of COPD, of asthma, of ARDS, and questionnaires, for example. A subtype or a subphenotype would be something like extrapulmonary ARDS or RA-associated ILD, a subtype of that phenotype. And then an endotype would be then a subtype with a shared biologic mechanism, which we're trying to get to now, like Th2 high asthma or Th2 high COPD or emphysema due to alpha-1 antitrypsin deficiency. And then a biomarker, which is relevant to all of this, is something that indicates normal biology, a disease process, or response to treatment. And in lung disease, we can think about blood tests, CAT scans, questionnaires, for example. I think everybody's familiar with the central dogma of molecular biology. You learned that in college and medical school, DNA, RNA, protein, and that sort of puts in a framework of what we're doing with the omics data. But just to remember, when we're talking about human disease epidemiology, it's much more complicated than that. Just from the biologic side, we have to think about things like epigenetic modifications of DNA, metabolites, and then thinking what's really driving our phenotypes. It's probably the proteins and the metabolites that are doing that. And then the environment in lung disease plays a huge interaction, both exposures, infections, allergies, for example. So just keeping all this in context as we're doing all of our genomic studies. So just some things to think about when we're doing human genetic epidemiology. We have to think, what's our study population in terms of the sample size, the racial and ethnic breakup of that, and I'd love to have a whole discussion of that, but maybe another future webinar we can do that. What our phenotypes actually are, are we using FEV1 in one study and FEV1% predicted in another study, or physician diagnosis or things like that for COPD, and how do we put those together? We'll get to things like disease heterogeneity. I think it's very important, and Stephanie mentioned that smoking status confounds a lot of our gene expression studies, and we have to be very careful to think about that in lung disease. And we'll talk a little bit about tissue, and Stephanie touched on that as well. So I just want to point you to these two papers that came out in the last couple of years, two reports from the ATS. One that I co-authored on use of high throughput sequencing, and this research really goes into a lot more detail on that. And then Ann Wu and Blanca Himes did a report on biomarkers in lung disease. So I refer you to both of those papers. So this is how I look for endotypes in COPD. Take a little bit different tack than Stephanie's group does. So first, as a clinician, I'd look for a clinical problem. And then I try to understand the epidemiology. So what are the phenotypes relevant to that clinical problem, and how can we define subtypes of disease? And then at the last step, how do we put in the genomics or the biomarkers to get to that endotyping? So this is a pretty obvious question. So are all COPD patients the same? If you look at these two CAT scans, one on the left with severe destructive emphysema, one on the right with severe airflow obstruction, but no emphysema and signs of airway disease on the scan, it's pretty obvious that all COPD patients aren't the same. And if we showed our colleagues in oncology that we actually treat both of these patients largely the same, they would think we're crazy. And that's really the motivation for a lot of this subtyping is we have different disease pathophysiologies, but for the most part, we treat them entirely the same in COPD. Just to remind you, this isn't new. It's been known for over 60 years. And you remember from your med school textbooks, the pink puffer and the blue bloater with some correspondence to emphysema and airway disease phenotypes in COPD. So most of my work has been in the COPD gene study, which is an observational study at 21 centers across the US. And starting in 2007, we enrolled over 10,000 smokers with and without COPD, and they've I've come back for 5-year or 10-year visits where we've done spirometry, questionnaire, 6-minute walk, and chest CT scans really to determine these phenotypes, as well as blood draws for multiple omics studies. So one of the things we did initially is looking at that emphysema airway predominant and said, well, what about the pink puffers and blue bloaters? How useful are they? And I apologize, my blue turned yellow when I made these slides, but we defined, based on sort of expert consensus, emphysema, weight, oxygen saturation, pink puffers and blue bloaters. And even in the most severe COPD, the GOLD 4, only about 10% of the patients met a definition of pink puffers, and very few met that blue bloater definition. So yes, it persists in textbooks, but probably not useful or relevant for current epidemiology. But that idea of airway versus emphysema predominant COPD is valuable, and we used CAT scans to define how much emphysema somebody had, and used cutoffs to make airway predominant group and emphysema predominant group, and still defining the clinical epidemiology, looked at other clinical features like asthma COPD overlap, more common in the airway predominant, bronchodilator responsiveness, more common in the airway predominant, things like exacerbations and chronic bronchitis, which you would think would be more of the airway predominant, really were not statistically different. But then comorbidities became very interesting. So higher prevalence of cardiovascular disease and emphysema predominant, but a higher prevalence of diabetes and the metabolic syndrome in the airway predominant COPD. So getting at some idea of systemic inflammation in these people. So what we've been using, like Stephanie's group, to do most of our omics has been RNA sequencing. And just to remember, compared to your genetics that you're born with, the gene expression profiling is context dependent. And what that means, it depends on your tissue, your disease state. And for that reason, it's a little more challenging to run, but also may more closely reflect the disease pathophysiology. And we can often do these in smaller sample sizes than the tens or hundreds of thousands needed for genetic studies. My group has been focusing primarily on blood RNA-seq in the COPD gene study. And as Stephanie mentioned, we don't routinely do bronchoscopies in routine COPD care, as opposed to our colleagues in oncology who almost always have a tissue sample to run their omics on. We have to use blood, and it may be better capturing the inflammatory phenotypes like the ones that Stephanie mentioned in her talk, and obviously can be applied in large-scale epidemiology studies. And at this point, we have in COPD gene about 4,000 subjects with blood RNA sequencing from that second visit at the 5-year follow-up. So just to taking that airway disease into the RNA sequencing, and this is work that was led by John Yun, looking for genes associated with a CT biomarker of airway disease, airway wall thickness, and found that most of the genes that we found in the blood were antiviral response genes or interferon signaling genes, which again, very similar to what you heard about in the last talk, and use those genes to create a gene expression score, which we found to be associated with lung function and with COPD exacerbations. Another clinical phenotype that we've been very interested in is eosinophilic COPD, and obviously there's multiple reasons why we're interested in that. It's part of the current GOLD guidelines. You see right here in Group D, we consider inhaled corticosteroids for people with elevated eosinophil counts over 300, and we'll get back to that in a second, and that there are anti-eosinophil drugs that are used in asthma that have been trialed with limited success in COPD, and it's possibly due to the definitions of subjects that have been used. So again, going back to the epidemiology, again, work led by John Yun, showing that in COPD gene, there's a linear increase in the exacerbation risk with increase in eosinophil count. But when we did ROC curve analysis, showed that that cutoff of 300 maybe had the highest area under the curve. And this was one of the papers that was, went into that cutoff in the GOLD guidelines, and just showing that clinical phenotype of eosinophilic COPD has some, but really not complete overlap with asthma COPD overlap, which we'll talk about in a second. So again, moving on to the RNA sequencing, found differentially expressed genes in eosinophilic COPD versus non-eosinophilic COPD. And very interestingly, again, like Stephanie's group, going to public data sets and finding overlap between our eosinophilic COPD genes in eosinophilic signature genes, eosinophilic asthma genes, and genes that are changed with response to benralizumab, one of the anti-IL-5 therapies. So like I mentioned in the title, just like Stephanie's group, we've been very interested in asthma COPD overlap. And just again, that thought originated with the Dutch hypothesis in 1961, just showing that there's really very few new ideas in pulmonary disease research. And they propose these common host factors for asthma and COPD, things like airway hyperresponsiveness and atopy. And those of us interested in genetics have said, well, maybe genetics is one of those common host factors. It's pretty common, up to 50%, depending on which studies you look at. And we all know clinically, these are the patients who have more symptoms, use more meds, show up in the hospital more and cost more money, and not just our clinical intuition, but there's been multiple large-scale studies that have borne that out. So we've looked at asthma COPD overlap in the COPD gene, and we use this clinical definition to find the phenotype. So smoking history, airflow obstruction, and then a self-report of a physician diagnosis of asthma before the age of 40 to define asthma COPD overlap. Found that 12% of the COPD patients had that overlap, more common in women and African Americans. Interestingly, there was a statistically significant difference in bronchodilator response, but it was 110 mL versus 90 mL, so not really clinically significant at all. And just like the previous database studies I mentioned, worse quality of life in asthma COPD overlap and higher exacerbation rates, and also other markers that led us to believe this is a real phenotype, like higher incidence of family history of asthma and personal history of hay fever. So went on to do a genome-wide association study in this phenotype, and this is something called the Manhattan plot, is how we display the results of millions of genetic variants on the x-axis across the chromosomes, and then the negative log p-value on the y-axis, and we're looking for skyscrapers. It doesn't show up really well here, but we can blow them up into these locus zoom plots, and this was our top association, was with a G protein receptor involved in eosinophil activation. Like I said before, we've been interested in biomarkers as well, and looking at IgE levels as a biomarker for asthma COPD overlap, and measured it in almost 3,000 subjects in COPD gene, and defined atopy by these pretty standard cutoffs, total IgE of 100, or at least one positive specific IgE for a panel of common allergens. And showed, here's our COPD subjects, here's our asthma COPD. So this here is the asthma COPD overlap, but really, only about 60% of the asthma COPD overlap were atopic. And we found that the people who are atopic asthma COPD overlap versus non-atopic asthma COPD overlap had different clinical phenotypes, which may be interesting, but overall IgE was not really a useful biomarker for asthma COPD overlap. And just to follow that up, we used, again, gone back to our blood gene expression and used the Th2 score that Stephanie's group had published on before in bronchial brushings, but we derived that now in blood, show that score is higher in asthma COPD overlap, associated with eosinophil counts, lower lung function, and higher exacerbations for that score that they derived in lung tissue that we can now use as a blood biomarker for these phenotypes. So just to wrap up here, this is what I call the grand challenge, is how do we take all these different phenotypes, and they're using endophenotypes here, meaning more of phenotypes or clusters, not how we're talking endotypes. I've talked about sort of one omics data type at a time, but how do we layer them all together and really come up with the endotypes or the biological clusters how they're described in this paper. And then once we get those biological clusters or endotypes, we can do a lot of different things. We can look at biomarkers for prediction, diagnosis, prognosis. We could select subgroups for clinical trials, and ideally we'll have a much more rational treatments, both in trials and in therapy for those subjects. And with that, I'll end. So thank you.
Great. Thanks so much, Craig. All right. I do see one question coming through asking whether relating to measures of transcription gene expression, could you describe what distinguishes RNA-seq, mRNA, miRNA from eQTL? Maybe a lot to get to, but.
Sure. So RNA-seq, depending how you set up your library preparation, you can measure mRNA, long non-coding RNAs, microRNAs, usually not all in the same experiment. So it's more than just mRNAs, which were traditionally measured by microarrays. So you have a broader range of both the mRNAs as well as the regulatory RNAs. And then what we mean by eQTL analysis is integrating two data sets. So looking at how genetic variants affect mRNA levels. That would be an eQTL. So what that is, it's a statistical association, but we think many of these may be biologically relevant, and then we can use those statistical associations between genetic variants and transcript levels to try to understand the genome-wide association or other genetic results. So what is the SNP or genetic variant doing? Could it be affecting the phenotype through affecting expression? So that's what we mean by eQTL analysis. And I didn't really get into the integrative except in the last slide. And I think that's, like I said, the challenge is how do we put more than one omics data type together with these clinical phenotypes to understand our diseases of interest.
Great, thank you. So now I'm going to introduce our last speaker, Pratik Sinha, who is at Washington University, and he's going to speak about inflammatory phenotypes in ARDS and COVID-19. Thanks, Pratik.
Hi, thank you, Carrie, for the introduction, and also thank you very much for inviting me to come and talk today. So I am going to talk to you today about finding homogeneous phenotypes in acutely injured lungs. I'm currently based as an assistant professor at Washington University, and the overview of my talk is that I'm going to take you through a whirlwind journey about the background of what we've been doing in terms of molecular phenotyping in ARDS. And then actually all of this is going to be a whirlwind because I see I'm quite pressed for time. But I'm then going to talk to you a little bit about how we've applied these phenotypes and sort them in COVID-19. And then I'm going to talk a little bit about the future direction that we intend to expand our research into. So what better place to start than ask the question, what is ARDS? And when you look at it from a clinical and a basic scientist standpoint, actually ARDS means lots of things to different people. Fundamentally, the problem comes from its definition. Currently, the Berlin definition is what we use, and this mandates that if you have a PF ratio of less than 300 millimeters of mercury, if your onset of symptoms is less than 7 days, you have bilateral opacification on your chest x-rays that's not fully explained by a cardiac cause or fluid overload, and you're getting a PEEP of 5, then by definition, you have ARDS. Now, these sets of conditions, you will agree, are frequently encountered in patients who are mechanically ventilated. So overall, the definition can be perceived as non-specific and really quite generic and arbitrary. And as a consequence, there is immense heterogeneity that is subsumed within this broad Berlin definition. And these raise its sort of ugly head in pathology, which seems to be inconsistent, whereas previously diffuse alveolar damage was the hallmark of ARDS pathology. Now it's only found in about 45% of these patients. And it's interesting to note that this divergence has only started since we introduced our first clinical definition, which was the AECC definition in 1994. And subsequently, there's etiological, radiological, clinical, and physiological heterogeneity in ARDS. And I'd like to point out, finally, that I'm very jealous of Stephanie and Craig, not only for their inherited brilliance, but also they get to study diseases which are kind of plodding and slow moving. Whereas the problem with studying critical care illnesses is what I call this a sort of temporal hostility. Our patients are incredibly unstable, and it's very difficult to know that the signals that you're studying are specific to that time point, or whether that instability of the patient needs to be factored in. So for those reasons, I'll come on to that in a little bit later. The question is, why is this then a problem? The real problem of this is not just in ARDS, but also sepsis, which is equally problematic in terms of its heterogeneity, is that the trials that we have done in the past have not been great. And this is a list, and this is only a tip of the iceberg of the pharmacotherapies that we have trialed in RCTs in ARDS. And for those of you who know the literature well, and I'll save you some reading, these are all negative. And as I said, this is just merely the tip of the iceberg. Most of the trials that we do in ARDS and sepsis are in fact negative. So our approach, and again, broadly talking about the definitions that everybody has spoken about, we describe our work as seeking molecular phenotypes. And our purview really, when we started this project, and emphasize that most of the early phenotyping work has been done by Carolyn Calfee, who's my mentor at UCSF. We wanted to use approaches that were unbiased, and because there is this issue of disease severity and fluctuating severity, we used a composite of clinical and biological data in our modeling to inform our phenotypes. In terms of the biological information, we used circulating protein biomarkers, and the clinical data we use were things like vital signs, demographics, and laboratory values. So that we're actually trying to incorporate what the patient's status is at the time that we did the modeling. And very much we were looking for multivariate solutions, because biomarkers and cytokines in critical care have been measured for a number of years, decades prior to this work, and really we've not been able to find a single cutoff. And it probably stands to reason that in this milieu of heterogeneity, it's highly improbable that we will find a single biomarker that can give us a cutoff that's useful. The other, of course, mandate was that whatever we identify needs to be robust, reproducible, and there needs to be some palpable clinical utility for this. And the work that I have mostly done with our group has been really to think a little bit about how we can implement these complex machine learning algorithms and data into the clinical setting. So to find our phenotypes, we use an algorithm called latent class analysis. This is a mathematical mixture modeling. You can view this as a probabilistic form of unsupervised clustering. And the idea is that in a given multivariate distribution, we assume that within this population, there are some hidden unmeasured subgroups. And the idea is that you use latent class models to you use lots of different variables within that model to try and identify unmeasured or hidden, that's the word latent comes from, latent measures, which are these subgroups with perforated lines. We have so far only published this in randomized controlled trials. For the model, we use pre-enrollment data or data from either at the time or preceding enrollment. So far, we have used latent class analysis in 5 randomized controlled trials. This includes the ARMA trial, which tested low versus high tidal volume, the alveoli trial, which tested high PEEP versus low PEEP, the FACT trial, which tested conservative versus liberal fluid management. Those 3 were NHLBI trials. This was a UK trial, which tested simvastatin versus placebo. And finally, most recently, we've tested it in the SAILS trial, which tested rosuvastatin therapy versus placebo. And all of these are ARDS trials. So consistently and independently across all 5 trials, we have identified 2 phenotypes in ARDS. We have termed these the hypo, which is represented by the blue line, and the hyperinflammatory phenotypes. And what you see here on the x-axis are the continuous variables that we used in the model. And on the y-axis are the mean standardized values for each of these continuous variables stratified by the 2 phenotypes. So in the areas where you can see the greatest departure between the hyper and the hypo-inflammatory phenotype, which is at both of these ends, these variables were the most important in terms of differentiating the two phenotypes. We have called the hyperinflammatory phenotype because it is associated with elevated levels of circulating biomarkers, including IL-6, IL-8, soluble TNFR1, PI1, and higher levels of creatinine and bilirubin, suggesting there is increased end organ dysfunction. At the other end, the hyperinflammatory phenotype was also associated with lower levels of bicarbonate, lower levels of protein C, lower platelets, lower systolic blood pressure, and albumin. It is also interesting to note that in terms of risk factors of ARDS, the hyperinflammatory phenotype was predominantly associated with sepsis of non-pulmonary origin, i.e. systemic sepsis, whereas the hypoinflammatory phenotype was more associated with direct causes of lung injury, including aspiration and bacterial pneumonia. In terms of composition, about 30% of our population was assigned the hyperinflammatory phenotype. And again, consistently across all 5 trials, the mortality in the hypoinflammatory phenotype was about 20%, and in the hyperinflammatory phenotype, this was twice that, so it was double that, ranging between 40 to 50%. And again, consistent across all 5 phenotypes. And I guess importantly, and this comes into perhaps one of the reasons why this work has been has got so much traction, but also demonstrating some of its potential clinical utility, is that in 3 of these trials we observed differential treatment responses within the phenotypes. In 2 of these trials, in PEEP, in alveoli, which tested high PEEP versus low PEEP, and in FACT, which tested conservative versus liberal fluid management, we observed a heterogeneous treatment effect with the randomized interventions and the phenotypes. In HARP2, what we noted was that in the hyperinflammatory phenotype, simvastatin was associated with a significant survival benefit compared to placebo, which was not observed either in the hypoinflammatory phenotype or in the original trial. Worth emphasizing again that all of these trials were negative, and also that these effects were not observed when we stratified the same population by other metrics of disease severity, such as PF ratio, Apache scores, suggesting that the information, both biological and in terms of these treatment effects, are specific to these phenotypes that we have identified. So at this point, we were like this chap here in the playground, we were stuck. And the question was, how do we implement these really complex models, which comprise of 30 or 40 variables, but more importantly, the variables that we put in order to fit in through some of the prior modeling assumptions, we transform the data so that they're standardized to the mean. So how do we identify models that are clinically viable? So the first solution was to try and develop more parsimonious models comprising of 3 to 4 variables. In order to do this, we used an array of machine learning algorithms, which we first used for variable selection. We identified 6 of the key variables that were common across these array of machine learning algorithms, and then we built parsimonious models, which we trained in a combined data set of ARMA, alveoli, and FACT, and we validated in the most contemporaneous data set, which was SAILS. The 3-variable model, which was the primary model, I'd probably emphasize here that if you go to the manuscript, there are a multitude of different models that we have described, which are a permutation of the top 6 variables. But for our primary model, we described IL-8, bicarbonate, and protein C as the 3-variable model, and the addition of vasopressor as the 4-variable model. And we found that the models performed with high accuracy with really good performance metrics with AUC of 0.94 and 0.95. And importantly, when we took one of these parsimonious models and applied it to the HARP2 data set, we found the same significant survival benefit in the hyperinflammatory phenotype with phenotypes identified using this approach as we did with the original latent class analysis models. However, we are still stuck, and it's important to maybe pause here and appreciate that there is a growing pandemic of grown men being stuck in children's playgrounds, which I hadn't quite realized until I prepared this talk. The reason why we were stuck here was that we have no point of care test, or at least no multivariate point of care test for biomarkers. So the next challenge we posed ourselves was, can we use readily available clinical data to try and identify these phenotypes at the bedside? Now, using traditional logistic regression models, the model performance was fairly mediocre, with AUCs of around 0.75 to 0.8. So we again turned to some more novel machine learning approaches. We used XGBoost, which is a form of gradient boosting machine. And what this is, these are recursive partitioning techniques. Essentially, you try and find solutions by partitioning the data, and it builds multiple trees into the model. And the beauty of this particular approach is that it learns from classification errors of the incumbent trees in the model and informs the new trees that are added on using these methods. And actually, this is a very popular method in people who use predictive modeling. Again, we use the same approach in terms of our derivation and validation data set. Also, what we did here was, as a form of cross-validation, we took permutations of the 4 cohorts, and it enabled us to cross-validate within these different cohorts to show robustness of the model. In terms of the data we used, broadly speaking, we broke them down into demographics, vital signs, respiratory variables, and laboratory tests. And what we found was that these classifier models also performed at a high accuracy. The performance metrics were quite high, with an AUC of 0.94 and 0.95 across the various permutations that we used. The clinical characteristics, including outcomes of the phenotypes identified using these machine learning clinical classifier models, were very similar to the LCA-derived phenotypes. The biological properties of the phenotypes were also quite similar. IL-8 levels were significantly higher in the hyperinflammatory phenotype compared to the hypoinflammatory phenotype. Conversely, protein C was lower in the hyperinflammatory phenotype compared to the hypoinflammatory phenotype. And these biological findings were consistent across all the biomarkers that we measured. And we obviously kept this abstracted from the model development and validation process. What's quite interesting is that the absolute median values and distribution of these protein biomarkers were very similar in the clinical classifier models compared to the original LCA-derived phenotypes. And again, it's important to emphasize that the heterogeneous treatment effects that we observed, both in FACT and in alveoli, we also observed using this clinical classifier models. We couldn't apply this to HARP2 because they only collected a very selected set of variables in HARP2, whereas in the NHLBI data sets, there were more complete data. So what about COVID-19? We asked ourselves the question, what is the prevalence of these ARDS phenotypes in COVID-19? Now, we applied the parsimonious model, and this is quite a cool study. What we did was we took a novel point of care testing developed by Randox, which is a diagnostic company in the UK, and we measured IL-6 soluble TNFR in real time using fresh blood, fresh plasma, as well as serum bicarbonate for real-time identification and classification of these phenotypes. Now, this is a very small study because of restrictions in terms of trying to analyze and do COVID research at the front end of the pandemic. So it comprises of only 39 patients. And again, this is published in the Lancet Respiratory Medicine recently. And our findings very briefly were that the prevalence of a hyperinflammatory phenotype in this population was actually quite low, 10 to 20%, compared to in a similar population of ARDS in all comers, it's about 35% in the UK. In the data set that we've studied. Interestingly, whilst the hyperinflammatory phenotype was significantly higher than the hypoinflammatory phenotype, the actual absolute 28-day mortality of the hypoinflammatory phenotype was approaching 40%, whereas in all our other data sets, this was about 20%. And also what we learned was that the circulating inflammatory responses in COVID-19 seems lower. The IL-6 levels were comparable, although we have some other data which shows that it is significantly lower in COVID-19 compared to regular ARDS. Soluble TNFR1 was significantly lower in this cohort compared to in COVID compared to a matched ARDS cohort. Yet the mortality seems quite high. So to us, this suggests that we need novel biomarkers that might inform new phenotypes. And the phenotypes that we've identified previously may not be applicable to COVID-19 ARDS, which is secondary to a single etiology. And we're working on this in terms of future directions. But also importantly, worth emphasizing that this technology and study demonstrates to us that real-time phenotyping is feasible. So to summarize the work we have done so far, we've identified 2 distinct phenotypes of ARDS using circulating protein biomarkers and a composite of clinical data. These are robust, reproducible. We've used clinically viable we've described clinically viable models, and really it's taken us from a pathway, a descriptive pipeline from discovery to clinical implementation, and it may offer a route to prognostic and predictive enrichment. So the future, we want to study the stability of these phenotypes over time and see what the interaction of this temporal instability is. And we want to question phenotype generalizability, whether it's generalizable to unselected populations of ARDS, which we've already studied, and I can tell you that it is generalizable. But another question is, are these phenotypes specific to ARDS, or can they be applied to other inflammatory critical care conditions? And we're studying that next. We need to get a deeper understanding of phenotypes, and we're going to be leaning more on transcriptomic data and in terms of whole blood and single cell sequencing. We're going to maybe we're getting to the point where we now need to start thinking about what the potential phenotype-based clinical trials might be. And the pertinent question here is, what are the trial candidates in terms of interventions to study these groups? And finally, I'll leave you with the question that do we still need these broad, non-specific definitions of clinical diagnoses such as ARDS and sepsis, and how helpful are they, and do they add to the uncertainty? I've got so many people I want to acknowledge. As you can imagine, this type of work is based on multiple collaborations. Specifically, I'd like to thank my mentor, Dr. Carolyn Calfee, and I would like to thank all the participants, family and patients who took part in these clinical trials and studies. And finally, I'm happy to take any questions.
Thanks, Pratik. I have one question coming in through the chat about what was the time frame of when the samples were taken in the COVID study? Was it earlier than the ARDS?
Yes, so that's a great question. It was within 48 hours from the diagnosis of ARDS. However, most of them were comparable to the other studies that we did. The protocol that we devised for this study was in line with replicating previous randomized controlled trials. So it's very comparable.
I saw that I think it was in the rosuvastatin data you were showing when the hyperinflammatory phenotype mortality curves separated quite early. And I didn't get a chance to see what the time scale was, but did you think that there's something rosuvastatin does in the first couple days?
Well, we did look at it, but I don't think there's anything there in the rosuvastatin. I have some inherent skepticism about the type of molecule that they used, which is rosuvastatin is a hydrophilic statin, whereas almost all of the data, both preclinical and clinical, are all in support of lipophilic statins. And it's quite interesting. There's a paper by Angela Rogers and her colleagues, which did some subgroup analysis that showed that there is a subset of patients in which rosuvastatin might actually cause harm, dependent on what their IL-18 levels are. So it wouldn't have been my drug of choice, rosuvastatin, even though it has a narrower sort of a toxicity profile than simvastatin or atorvastatin. There really is no, even in animal models, the putative anti-inflammatory benefits of statins are only observed in lipophilic molecules and not in rosuvastatin. So I have some grave doubts about it.
Okay. Thank you. This is so interesting. Thanks for tying COVID into it too, to make it really highly relevant for all of us. So
yeah, pleasure.
Thanks to all the presenters and everybody for joining us. If you have a moment to give feedback, that would be helpful for us moving forward. So thank you.
I'm going to end the meeting now.
Great. Thanks, Liz.
Thank you, everybody.
Bye.
Bye.